In this video, we will discuss about SERCA forward pumping cycle and some of the points that affects this cycle. This SERCA forward pumping cycle is usually occurs in muscle cell, and this SERCA proteins is found in sarcoplasmic reticulum, on the membrane of sarcoplasmic reticulum. So what happens when the muscle contracts is the protein that is present within the sarcoplasmic reticulum is released, and that is compensated by protonated ions. And at this time, the sarcoplasmic protein is present in E2 state, and in E2 state within the sarcoplasmic lumen. And however, this loss of Ca2 plus or this release of Ca2 plus need to be compensated, and this can be compensated by the ATP. This can be compensated by the calcium that is present in the cytoplasm, cytosol of the cell. And in order to transport the proteins from the cytosol to the sarcoplasmic lumen, there is a conformation change in the sarcoplasmic SERCA protein, or we can say sarcoplasmic and sarcoendoplasmic reticulum. I'm not able to recall this full form, but SERCA protein, let's say that there is a change in the conformation of SERCA protein from E2 to E1 when ATP is bound to it, and that helps to exchange these protonated ions with Ca2 plus ion. And this E1 conformation has Ca2 plus ions, and ATP is also attached to it, and with the hydrolysis of ATP, the Ca2 plus will be transported across this sarcoplasmic membrane into the lumen. And one more thing that we need to understand, that the concentration of free Ca2 within the cytosol and within the sarcoplasmic lumen is completely different. So this transfer that has occurred is completely against the driving force, as the concentration of Ca2 plus in the sarcoplasmic lumen is around 10,000 times more than the Ca2 plus that is present within the cytosol of the cell. And there are a few other points that we need to understand that affect this SERCA cycle. One of them is phospholamban. So what this phospholamban then is, when it binds to the SERCA protein, it makes the SERCA proteins ineffective, not that much effective as it is when it is free from phospholamban. So it helps in controlling the rate of Ca2 plus movement across the sarcoplasmic membrane. And another thing, another protein is this calsequestrin. What calsequestrin does is it binds to the free Ca2 plus that is present in the sarcoplasmic lumen. And what happens because of this? The gradient, that is gradient of free Ca2 plus that is present in the between the sarcoplasmic lumen and the cytosol is reduced compared to a situation where there is no calsequestrin present in the sarcoplasmic lumen. So this helps in reducing the gradient, and that finally helps in less energy required to transport Ca2 plus from cytosol to sarcoplasmic lumen, as less work needs to be done against the driving force, as the driving force uh, is reduced in case of calsequestrin present in the sarcoplasmic lumen. And in order to study this SERCA forward pumping cycle, this it has to be artificially controlled, and this artificially control has been done by Thapis Gargin and Istarksomine. So what this Thapis Gargin does, it inhibit the function of SERCA proteins, and this isotach uh, taroxamine, it accelerate the function of SERCA protein. And one of the study that has been frequently, or that has been done to understand the effect of ER stress by inducing artificially inducing ER stress by inhibiting this SERCA cycle, and that has been done by Thapis Gargin, by the addition of Thapis Gargin in the cells. So with this, the normal function of the sarcoplasmic lumen has been affected, and which result in generation of ER stress, and that can be monitored by various signals. Some of them is ATF4 signal, and another one is ATF6 signal, and third one is XPV1 signal that we have already covered during ER stress video. So that's all for this video. Thank you for your time.